Hi, hello, namaste, good evening to all. Myself Murali PG. Today let us have the YouTube class on Biotechnology Principle and Process chapter under Biotechnology unit. In the next class I will going to take Biotechnology and its application. Okay. Today let us have Biotechnology Principle and Process. So Unacademy is one of the good platform to achieve the goal in your oncoming Karnataka CET. With respect to that one, we are with the some of the important courses for you. Some of the important courses for you. So plus a course where we are going to have the Karnataka CET aspirants. So one who are having that particular year Karnataka CET 2021 aspirants, and here's 12 month, 6 month and 1 month plan will be there. And with respect to this one, we are having following features. And these features are as follows with the complete theory, MCQ, doubt clearance and mega quiz. So once you subscribe for this one, you will get unlimited access to all the educators topic, and use my referral code or use code KCET Murali and get 10% off with respect to this price values. Okay, so that is the. And secondly, it is a comprehensive batch. We introduced this batch from last 2 months, and it is also coming under a plus course. What is the importance of this one? Here the features are as follows: complete syllabus, tips and tricks by top educators, topic wise practice questions, and full length mock test will be there. And here also you can use my referral code or use code and get 10% off with respect to the price value. Myself, I am taking this one under Karnataka CET comprehensive batch English Kannada version. Along with me, the 3 other respected educators will going to take their respective topics. And the newly introducing course for this month with respect to 15th of December, it is the Masters batch for Karnataka CET KCET 2021, and myself the biology, and Kalimullah Sharif Saab for mathematics. Okay, yes, yes, welcome to the class, namaste. And this batch will going to start from tomorrow onwards, 15th of December. Okay. Then comprehensive batch for Karnataka CET. This was first started from 1st of September. Myself, I am taking this comprehensive batch also. Then best plans for Karnataka CET aspirants, where 12-month plan, 6-month plan is there, and 6-month full syllabus, dedicated doubt clearing sessions, course completion in Kannada language, and no cost EMI available on 6-month and above subscription plan, multiple batches launches every month. So that will helpful for you regarding with our courses. Then best plan for Karnataka CET 2021, and for that multiple batches launching every month. So make a note of it, and use my referral code KCET Murali and get 10% off with respect to all these plus batches. And if you want more from my side, please use this opportunity with respect to the launching, with respect to joining of this launching batches. Okay. Then daily T20 free practice test for Karnataka CET. This is a new things, and attempt the test for free. So free purpose, under free basis you will get the concept. Okay. And champion batch, so it was created started from 15th October, like that of comprehensive batch. Here also you will get the PCM, PCMB and PCB batches. Okay, thank you for this one, and thank you everybody. So welcome to class, and use the opportunity of these classes and get good things from our side. So as a result, you will get the good marks and good achievement in oncoming Karnataka CET. So let us crack the CET together and have a good day. Okay, now let us start our discussion of questions regarding with biotechnology, its principle and process. So first question here. The enzyme that cuts specifically recognition site in the DNA is known as what? So there are enzymes involved in various different types of recombinant DNA technology and biotechnology process. So out of this, which enzyme that cut specifically recognition site in the DNA sequence, which is that one? So the first question answer is D. D is the right answer. Okay, D is the right answer. Restriction endonucleases that will going to cut the DNA at specific site. Yes, Sagarappa Karjol, welcome to the class. Namaste, good evening. Yes, Saragappa, for the first question it is D. Okay, it is not A. A for the joining purpose. Ligase refers to joining of DNA. It is for joining. Okay, DNA polymerase, it is for what? DNA replication. DNA dependent DNA polymerase, it is for replication. And reverse transcription, transcriptase for RNA to give DNA. That is for reverse transcriptase. From RNA we are going to get DNA. For that, reverse transcriptase enzyme is going to help. Got it? So right answer is D for first question, Sargappa. And second one, DNA can be introduced into any cell by which method? So artificial type or artificial method of introduction of DNA. So by using which method we can introduce the DNA into any cell? That is micro injection. Injection means micro injection. Being complexed with the calcium salt, gel electrophoresis, being placed along with the cell. So the right answer for this one, it is injection. Refers to micro injection. Okay. So micro injection can be utilized for the purpose of injecting the DNA into any cell. So as a result of that, we are going to get our desired character in the oncoming plant or in that plant. Okay. Yes, Shrishika, welcome to the class. Good evening. So now we are in second question. The right answer is micro injection is the right answer for question number second. Third question. Ability of a plant or animal cell to repeatedly divide and differentiate into complete organism is called what? What is the meaning of that process or the method? So ability of plant or animal cell to repeatedly divide, to undergo mitosis. For mitosis and differentiation will be there, and as a result we will going to get a complete organism. So for that purpose the plant or animal cell should have one particular character. Which is that character out of this option? Cloning, DNA fingerprinting, cellular totipotency and mitosis. So the right answer is C, mitosis. Sorry, right answer is C, cellular totipotency. Cellular totipotency, yes Saragappa, you are right. Cellular totipotency. So totipotency is the nature of the cells that will going to give entire organism after repeated mitosis and differentiation. So right answer for the third question, it is C. Sheshika, you are also right. Okay, let us move on to the next question. Restriction endonuclease is also known as what? What is the another term for restriction endonuclease? So DNA ligase, molecular glue, DNA polymerase, molecular seizure. Which is the right one? Yes, Sheshika, you are right for the previous question, cellular totipotency. So for the fourth question, the right answer is molecular seizures. So restriction endonucleases are called molecular seizures because they will going to cut the DNA in particular required region of the DNA sequence. Molecular glue refers to what? It will go into ligase or attachment, that is by DNA ligase. Molecular glue, DNA ligase. DNA polymerase, it is for replication. I said it in previous one question, isn't it? So right answer for fourth one, it is D. Extra chromosomal small circular double stranded DNA molecule in a bacterial cell is what? What is the terminology for this one? Extra chromosomal small circular double stranded DNA molecule in a bacterial cell is called plastid, plasmid, mitochondrion, chloroplast. It is called plastid. So plasmid. It is called plasmid. So plasmids are required, or it is going to be utilized in various recombinant DNA technology under biotechnological principle and process. So if you have any doubt, you can ask, okay, through our chat box. You are welcome. And next question number 6. I think I added 30 questions here. Yeah. Next question number 6. Introduction of foreign gene into plant or animal cell using micropipette. Micropipette, it is not the injection. Micropipette is termed as what? What is the terminology? It is called chemical mediated gene transfer. Foreign gene is going to be introduced into plant. So foreign gene means the gene of our interest, required gene, is introduced into the plant or animal cell using micropipette, that is called chemical mediated gene transfer methodology. Okay, it's not a particle gun or by using a gene gun. Their micro injection not, electroporation is not the right one. So right one is B. Okay. And next question number 7. Which one of the following is related with the genetic engineering? Genetic engineering. My dear students, am I audible clearly? Which one of the following is related with the genetic engineering? So 7th one, mutations, should be mutations, okay, MUTA, mutations, ribosomes, mitochondria, plasmid. It is nothing but D, plasmid. Plasmids are related with the genetic engineering. Okay, plasmids are related with the genetic engineering. In bacteria, genes for antibiotic resistance are usually located in which part? Plasmid, cytoplasm, plasm, mitochondria, nucleus. So genes for antibiotic resistance, that is very much essential for the bifurcation or differentiation of transferred cells or non-transferred. So as a result of this one, we can isolate the bacteria which is having the introduced new gene or not by using antibiotic resistance gene. So we can separate out the two types of bacteria, whether the introduced gene is working or not. To do that one, we are using this antibiotic resistance property phenomenon. So in bacteria, genes for antibiotic resistance are usually located in plasmid, cytoplasm, mitochondria, nucleus. So right answer for 8th one, it is the what? Plasmid, isn't it? In the plasmid we can observe that one. Next, a technique used to make numerous copies of specific segment of DNA quickly and accurately, that is by which method? So 9th one, it is called PCR, polymerase chain reaction. We can write PCR, polymerase chain reaction, isn't it? Polymerase chain reaction or PCR is the right answer. So where we can get multiple copies of required gene. Even though the sample DNA is very small, we can get the multiple copies of that DNA by using PCR. Then 10th question. Question number 10. Yes, Saurabha, welcome. Okay. Question number 10. The enzyme that cleaves DNA at a specific site producing sticky end is called what? Producing sticky end. It is called what? So the DNA is going to be sliced. So if the DNA strand is very large, DNA strand is very large or a very big DNA strand is present, and here we can cut the DNA molecule at a specific site by using the enzyme called restriction endonuclease at a particular site. So as a result of that, we are getting the DNA sequence with the sticky end. Sticky end means the part of the strand is not having the complementary base. A small part of the strand is not having the complementary base here. So it looks like this after cutting. Yes, like this. Here the complementarity is there, but when you are coming here, this is the sticky end because of the cutting of the DNA strand by restriction endonuclease. Yes, Shishika, for 9th one it is D. You are right. For yes, that is PCR, isn't it? Yeah. For 9th one, yeah, PCR, polymerase chain reaction. Can anyone give the name who developed this PCR? Can anyone give the name who developed this PCR? PCR is the technology. It was given by whom? Or technique, it was invented by whom? Anyone knows? Who invented the PCR? Yes, Lahari, you are right. For 10th question, it is A. Yes, I am asking about 9th question. Who invented PCR? Anyone can answer? Do you know? Yes, it is Carey Mullis. Carey Mullis developed PCR in 1980. In 1980, Carey Mullis developed PCR, and for his invention in 1992 he got Nobel Prize. He got Nobel Prize. Okay, that is a PCR. And for the next question, 10th question, the enzyme that cleaves the DNA at a specific site, that is called restriction endonuclease. Molecular seizures that will going to give the sticky ends in both strand, isn't it? So right answer for 10th one, it is A. Let us move on to the next question. 11th one, which of the following is a genetic vector? So genetic vector is important for carrying of the desired gene into the host cell. Which is that one? Phage, cosmid, plasmid. Which is the right one? Phage particle, cosmid particle, plasmid particle. Which is the right one for 11th question? Anyone? Yes, for next question, 11th one, which is the right answer? Which of the following is a genetic vector? Nobody knows. It is the plasmid. Generally it is called plasmid. Okay. Next question number 12. Restriction endonuclease are used in the genetic engineering because. Why we have to use the restriction endonuclease? What is the speciality? So the specialty is they can cut DNA at specific base sequences. Because of this reason, we are using DNA in the DNA experiment, recombinant DNA technology, restriction, endonucleases are going to be utilized, isn't it? Next question number 13. Ideal host for amplification of DNA molecule is amplification of DNA molecule. Which is that host? It is bacteria, or you can say E. coli. E. coli bacteria. So it is yes, Sarakappa, you are right. Cut DNA for the previous question. And for 13th question, C is the right answer. Ideal host for amplification of DNA molecule, it is by bacteria, E. coli molecule. Yes, Lahari, it is right. C is the right one. For 12th one also C. For 13th question, it is the C is the right answer. Okay. Then 14th one. TI plasmid naturally occurs in which organism or cell we can observe tumor inducing plasmid, TI plasmid, tumor inducing plasmid, Agrobacterium, Corynebacterium, Staphylococcus vibrio. So right answer, it is A, Agrobacterium tumefacium, isn't it? Agrobacterium tumefacium is the right answer, where we can observe TI plasmid. So it is going to be utilized for the introduction of gene into plant. Usually they will going to cause the cancerous growth, cyst formation in the plant. Along with that, we can introduce our interested gene, isn't it? So tumor inducing plasmid from the Agrobacterium tumefacium. It is one of the bacteria. So 15th question. Next question number 15. The sticky ends of fragmented DNA molecules are made up of what? In one of the previous question, I said which is going to produce a sticky end, that is a restriction endonuclease. Now, what about the chemical nature of that sticky ends of fragmented DNA molecule? It is made up of what? It is made up of methyl group. Methyl groups are maximum, where it is going to have the formation of sticky end after the action of restriction endonuclease. Next question number 16. Which of the following are the essential requirement for recombination? For recombinant DNA technology, which are the important things? That is single stranded DNA, DNA ligase, DNA polymerase 1, all of the above. It is nothing but DNA ligase, or it is called molecular glue, isn't it? Molecular glue. So it will going to join the two strands. It will going to join the two strands, isn't it? So right answer, it is B for 16th one. Okay, Saragappa, it is not I think it is not D. Okay, it is B. Okay, it is not D, it is D or B. Okay, it is not D, it is not all of the above. For the recombination technology, we want DNA ligase, because for the joining of foreign DNA particle with that of vector DNA. Then the plasmid derived from E. coli is the plasmid which is developed from E. coli, PBr 322, isn't it? PBr 322. So we can write this one as like this, P. Yeah, it's okay. PBr 322. So superfix and subsuffix like that we can write. Okay, don't worry. ಬರೆಯುವಂಥ ವಿಧಾನ ಉಂಟು, ಬಟ್ ಈಗ ಬರೆದ್ರು ಓಕೆ, ನಾನು ಜಸ್ಟ್ ಬರೆದಷ್ಟೇ ಇಲ್ಲಿ. ಸೊ PBr 3, just I written here. Okay, if you have any doubt you can ask. Okay, don't sit fight. So PBr 322 ಅದನ್ನು ಹೀಗೂ ವಿಧಾನ ಒಟ್ಟು ಬರ್ತೀನಿ. ಸೊ I written in a continuous manner, but we can write Actually it is the PBR322. Okay, so it is a E. coli cloning vector created in 1977 in the laboratory of Herbert Boyer at the University of California, San Francisco. Okay, because of that one it was named as PBR322. So we can write like this. Next question number 18. Am I right? Yeah, 18th question. Yes, for 17, Sir Devesh, welcome to the class. It is 17B is the right answer. Yeah. And for 18th one, TI plasmid is useful in which method? It is bringing tumor cells into plant cell to nearly any site on a chromosome, bringing new genes into plant cell, bringing new genes into animal cell. So TI plasmid is useful in which process? That is bringing new genes into plant cell. So to introduce the gene into plant cell, we can use a TI plasmid. So the right answer for 18th one, it is B. Then 19th question. Next question number 19. Yeah. Many copies of DNA molecule in a test tube are procured by which method? That is nothing but polymerase chain reaction, PCR. It is not molecular chain reaction or epimeral chain reaction. It is PCR, polymerase chain reaction. Right answer is A for this question. Yes, so for 19th question, the right answer it is A, polymerase chain reaction. Then BAM H1 Eco R1 Sal 1 are the type of what? Restriction endonuclease, restriction endoexidase, then restriction exonucleases, restriction polymerases. Which is the right one? Restriction endonucleases. Different types of restriction endonuclease. Example: BAM H1 Eco R1 Sal 1, etc. Okay. Next, 21st question. Retrovirus have genetic material which is, or retrovirus is made up of which kind of genetic material, or having which kind of genetic material? That is RNA. If you take HIV, it is a retrovirus, where RNA is the chief genetic material in the case of HIV. Even in the coronavirus, it is having RNA as a genetic material. Okay. Genetic engineering is possible because of what reason? Out of 22nd in that, out of 4 option, which is the right one? Here the right answer is the phenomenon of transduction in bacteria is well understood. We can see DNA by electron microscope. We can cut the DNA at a specific site by endonuclease like DNase 1. Restriction endonuclease purified from bacteria can be used in vitro. So genetic engineering is possible because of what reason? So the right answer here, genetic engineering, that is rDNA technology, is restriction endonuclease purified from bacteria can be used in vitro. In the laboratory condition, we can use this restriction endonuclease purified from bacteria cell, and this can be utilized in vitro or in the laboratory condition. Because of that reason, we can say it is a D is the right answer. Then plasmids are the suitable vectors for genetic cloning ants. Because of what reason? Plasmids. So plasmids are additional genetic material, isn't it? So they are indispensable. They are self-replicating units. They are essential for bacterial reproduction. None of the above. So 23, it is they are self-replicating unit, like that of main genetic material. They will undergo self-replication. Because of that reason, the plasmids which are incorporated with our foreign gene is going to be entering, or it is introduced into the host cell. So because of that reason of self-replicating units or self-replication phenomena, along with the host genetic material replication, the plasmid genetic material will also undergo replication. So genetic material is considered as genetic material, or any biomolecule is considered as genetic material when they are undergoing self-replication, isn't it? So it is having that phenomena. So plasmids are acting like a vector to carry the desired gene into host cell. Okay. Then 24th, which of the following is used in genetic engineering? Restriction endonuclease, mycobacterium, entamoeba. It is entamoeba. Okay. Entamoeba, B, entamoeba and pepsin. So right answer for 24th, it is A, restriction endonuclease. Yes, Devesh. For previous question, it is B. Okay, 23rd. 24th, A is the right answer. 25th, the first hormone artificially produced by culturing bacteria is which one? 25th, it is nothing but insulin. So insulin chain are going to be developed separately, and later it was joined together, and the right thing, it is the insulin by which the hormones are produced in the artificial way. In the case of bacteria, by using bioreactor, we can achieve this one. So right answer is A for 25th question. 26th, when the number of genes increases in response to some signal, the effect is called what? Number of genes increases in response to some signal. What is that? It is called gene amplification phenomenon. Gene amplification phenomenon. Okay. So thank you, Saurabh Patil. Yes, Saurabh Patil, it is 25th. Insulin is the right answer. You are right. And next question number 27. Which one of the following pair is correctly matched? RNA polymerase, RNA primer, restriction enzymes, genetic engineering, central dogma, codon, Okazaki fragment, splicing. It is correctly matched here. Restriction enzyme, that is genetic engineering, is the right one. Okay. Then plasmids are autonomously replicating mini chromosomes found in where? So plasmids, it is observed in Escherichia coli bacteria, isn't it? In Escherichia coli bacteria, we can observe the plasmids. So in the case of Escherichia coli, we can observe the plasmids. So right answer, it is C is the right answer for question number 28. For 27, it is B is the right answer. Saragappa, you are right. And for 28, it is C is the right answer. You are right. Yes, gene amplification. Next, 29th question. Another one question is remaining. Let us continue. Improvement of genotype of an organism by addition of some foreign gene is called what? Improvement of the thing. Genetic diversity, gene handling, and tissue culture, genetic engineering. This is called genetic engineering. Genetic engineering is the right one. And last question. Which bacteria found to be very useful in genetic engineering experiments are? So out of the following four options, which bacteria pair is going to be utilized in a maximum level? It is the Escherichia and Agrobacterium. E. coli and Agrobacterium tumefaciens. Isn't E. coli and Agrobacterium, Escherichia coli and Agrobacterium tumefaciens is the right one. Devesh, for the previous one, it is right. Sarakappa, you are right. Genetic engineering. Yes. So this is the last question. Escherichia coli and Agrobacterium tumefaciens are utilized in the maximum level. So Escherichia coli is having the plasmid. So the plasmid is introduced into the Escherichia coli with the desired gene. Agrobacterium tumefaciens is going to be utilized for the introduction of gene into plant cell. So it is very important thing. So by this I completed all the questions, and this is the Unacademy Plus subscription promotion slide. So if you want more, use this one. And how to get the entry into our special batch, special class. I am taking many special classes every month, and you go to you go to Unacademy Learners app from the Play Store, install the Unacademy Learners app or student app, and here under this you select Karnataka CET, and under Karnataka CET you search my name Murali PG, or use my code KCET Murali. Okay, KCET Murali or Murali PG name under biology section, and use this KCET Murali and going to so you are going to introduce it into, or you are incoming, or you are going to be there in the section under special class. Once you get into that, then you will going to have all the notification regarding with my special class if you followed me in Unacademy Learners app or student app. How to enter that one? Select the Unacademy Student Learner app from the Play Store, install it, and go to the Karnataka CET section and use my code KCET Murali or use my name Murali PG. Under biology you will get the entry. Okay, so that's why I'm telling. So use that special classes, that is also free. And if you want more things from my side, use the plus course where detailed theory, MCQ, doubt clearance will be there. So these are the comprehensive batch plus course. So next course batch is going to be there from tomorrow onwards under plus course. You can have these batches. Okay, you can join to this batch, comprehensive batch. Then good plans for Karnataka CET aspirants 2021, and this is for daily T20 free practice test for Karnataka CET. So these are there in under Unacademy Learners app. So please use this app, that is Learners app, and entry into Karnataka CET. Use my code, that is Murali KCET Murali, and get 10% off with respect to plus subscription for all the courses. So this is my request with you, and as a result you will get more things from our side, from Unacademy, from my side, and you can achieve a good goal, good things in the oncoming Karnataka CET. So let's crack the Karnataka CET together. Thank you one and all. Good night. I will going to wind up this session now. Thank you one and all.